Good morning, one and all. Myself, Dr. Rupasri, consultant obstetrician and gynecologist, Shrestha Hospital, Hyderabad. Today, we are going to see the mechanisms which help in transfer of substances across the placenta. So, what are the mechanisms? What are there is first point. It might be through simple diffusion. Okay. Second, it might be through facilitated diffusion. Third, it might be through active transfer. It might be through endocytosis. It might be through exocytosis. It might be through leakage. So, these are the six mechanisms through which the substances are transferred across the placenta. Okay, now we will come in detail. Okay, first one. As we have studied, first one is the simple diffusion. In simple diffusion, it is along the concentration gradient. Along the concentration gradient. Okay. So see, here the number of molecules are more. Okay. So higher the concentration. So along the concentration, what happens is we simple diffusion, the molecules come down from high concentration to the low concentration. It does not require any energy. Okay, any transporter molecule, nothing. Simply from high concentration to the low concentration. That is through simple diffusion. Okay. Now, the second is facilitated diffusion. In facilitated diffusion, they require an transporter protein. See, here is the high amount of concentration. To enter into low concentration, one transporter molecule or a protein is required. For example, if from the mother, if there is a requirement of glucose to transfer to the fetus, what happens is it utilizes the facilitated diffusion. In this, what happens is it utilizes a transporter protein known as GLUT1. Because of this transporter protein only, the amount of glucose at the higher concentration in the maternal blood enters into the fetal blood. Okay. So, this is the simple diffusion and this is the facilitated diffusion. Next, other thing is endocytosis. Third point is endocytosis. In this, what happens is, see, these are the extracellular molecules. What happens? In this, the cell membrane gets invaginated. Okay, this get invaginated and next it forms a vesicle. Okay, now the amount of extracellular molecules which are present outside the cell, it enters inside the cell, inside the vesicle. This is the process of endocytosis. The molecules which are present outside the membrane, it forms a invagination, it forms a vesicle, and now it enters into the inside the cell. This is endocytosis. Mostly amoeba. Amoeba takes its nutrition through this process only, phagocytosis. Phagocytosis. So this is the process of endocytosis. It forms a vesicle. Okay. And next is the fourth one is the exocytosis. See, in exocytosis, what happens is the amount of molecules which are present inside the vesicle breaks. Okay. So the amount which is present inside the vesicle now exits outside. This enters into the external membrane. Okay, so it breaks, the vesicle breaks and the amount of content which is present in the vesicle enter outside. This is the exocytosis. For example, mostly immunoglobulins. Immunoglobulins like IgG, IgM, what happens is it enters from the mother through the process of endocytosis. After entering into the fetus, it becomes exocytosis. Okay, initially there is a process of endocytosis. It enters the vesicle. From now by cycle, it enters the fetus through the process of exocytosis. Okay. So first we have learned simple diffusion, facilitated diffusion, endocytosis, exocytosis, and next is the active transfer. Now the fifth is active transfer. Active transfer means it is opposite to the concentration gradient. Okay, against the concentration gradient. Against the concentration gradient. See here, the number of atoms or the molecules are very less here. But by using an proton pump and energy transferring enzyme, okay, what happens is the amount which is very low or very less enter into the higher side. Okay, for example, okay, most amount of amino acids enter from the mother to the fetus through this active transfer manner by using an energy taking enzyme, that is ATPase. ATPase. Second, some of the electrolytes like calcium, okay, phosphorus, iron, they also enter from the mother to the fetus using this active transfer mechanism only. Okay. And now the last is sixth one, that is leakage. See, leakage is nothing. There is a rupture in the amniotic membrane. Okay, the amount of amniotic fluid comes out. That is the leakage. Okay, there is a rupture of the amniotic membrane and the amniotic fluid comes up. That is simply leakage. Okay, this is an example of premature rupture of membranes. PROM. Okay, so these are the six mechanisms by which the amount of substances or anything enter from the mother to the fetus. Okay, thank you.